So we are live on YouTube now. Good morning, welcome everyone. This session is being brought to you. This is the second part of Mendel and Heredity, which we are bringing as part of centenary second centenary celebrations of Mendel's birthday. And this session is being brought to you by the Joy of Learning Foundation in collaboration with the Luca magazine of KSSP, and this is being supported financially by Indian Oil Corporation Limited. Our resource people today are Dr. Swapna Khare and Apurva Bhatia. So I request Swapna ma'am to please start.
Okay, good morning everyone. Let's begin. So yesterday we started discussing how Mendel basically figured out how characters are transferred from parents to offspring. So this is just a brief recap of what we did yesterday. So what we saw is, first thing that Mendel did was he created true breeding species. That is, they had the character completely. They didn't have any variation. So he took different 7 characters. So example shown here is that he took purple flowered plant and white flowered pea plant. And when he did cross pollination and made hybrids, all of them only retained one trait, that is purple flower. So this was very, very surprising to him. So he wanted to see what happened to the white flowers. And when he allowed them to self-pollinate and looked at the next generation, which we call it as F2 generation, he actually got back the trait that was hiding. So this was the surprising part. So he got, and we all know that he got 3:1 ratio of purple to white, or tall or short. So every character that he was studying, he got the similar ratio. So he knew that there was something underlying this phenomena. So he wanted to explain this. So if all of you have understood this, we did a matchstick activity also. We are going to do one more activity today. So let's begin. Unless anyone has a question in their mind that you want to ask, otherwise we'll begin straight away. Should we start?
I think yes, because
yes, okay.
one person said yes. Yeah.
So Mendel then actually sat with all the data that he had collected, and he wanted to see what was happening. Just give me 1 second. Okay. So what he So he actually came up with different conclusions, and all of these are called as, initially were called as postulates, and now they are called as laws, after we have found that the same thing is valid for more systems. So the first thing he thought or inferred from his data is all the traits that are seen in a particular organism, they are controlled by some type of a factor. And these factors are passed unchanged from parents to offsprings. Now there are 2 very important things here that he thought about, which no one had thought of before. First is, he was one of the first ones to see that actually there is something particle-like which is actually responsible for the traits. Because yesterday we also saw that the current theory that time was that there is a blending of parent and both the parents. So he was the first one to say that there is something particulate factor that is responsible for a trait, and these factors are passing unchanged from parent to offspring. Now this is very important because what he saw was one character that disappears in F1 hybrid actually reappeared. So it was just hiding, nothing had happened to it. So these were the 2 important parts of his first statement, that is, traits are controlled by factors and factors are passed unchanged from parent to offspring. Now that we have moved on, we have actually changed the name. We don't call factors factors anymore. We call them genes. Okay. So hereafter, we will not use the word factor. We will use the word genes. So what he concluded is that each trait that you saw, whether it is a purple color or white colored petals, all of these traits that we discussed yesterday in the seed, they are actually controlled by a pair of genes. Okay, and these are the ones that we don't see, we only see their effect. So all traits are controlled by genes which are hidden from us. So now how many genes does an organism have or a plant have? So further he went and he said each plant must have a pair of genes for each character. Now this pair, that is, we are now talking about two genes, we call them now alleles, and I will explain you what is actually an allele. So now what we have genes which control every trait, and we have pair of genes for every character. So since we have two alleles, both the alleles can be either same or different. Let's look at it in a different way. So let's take the example of a short plant and tall plant. So if we have the true breeding tall plant, we will have both the genes or both alleles will be tall, which we denote by capital letter. Then the pure breeding short plant would have both alleles as the short alleles, which we denote by small t. So they will have small t and small t. And when we created a hybrid, since one allele came from one parent and the other from the other parent, the hybrid would have one tall allele and one short allele. So every trait that we see in any organism is controlled by different versions, which we call as allele. Okay, so there will be a allele for purple color, which we will call as capital P. There will be an allele which is responsible for the white colored flower, which we'll call as small p. And same for tall and short. For tall plant, the allele will be denoted by capital T, and the short allele will be denoted by small t. So plant height is controlled by a gene pair, tall allele or short allele, and flower color is controlled by a pair of alleles, capital P allele for purple color and small p allele for white. Okay, so now when you actually look at all the plants that we talked about yesterday, we talked about the parents which were pure breeding. So since they were pure breeding, they must have had capital P, capital P. So there are, when you actually have two alleles, you can have only three combinations. So this is capital P, that is both the alleles are same. This will be the true tall plant, or both the alleles in a plant are small p, that is short alleles, making this plant short one, or the one that we created as a hybrid, where one allele came from this parent and one allele came from this parent, creating small p, capital P hybrid. Okay, so for every gene pair, when you have two alleles, we create three combinations. But now the funny thing is, this is very easy to understand. This is very easy to understand. But now you have two different forms of alleles in single organism. So who actually, what happens? Who will show their effect? So then he actually concluded that this is actually his Mendel's second postulate, that when you have two different alleles in a single cell or in a single organism, what you see is one is dominant over the other. That is, one shows its effect, the second one cannot show its effect, it is hiding, its effect is not at all seen. So this trait which cannot show its effect, and it is really, if you see it as hiding or missing in case of F1 hybrid. So in F1 hybrid, when you had capital T and small t, the plant was tall simply because this allele could not show its effect when capital T was present. So one allele is dominant over the other. So the same phenomena should hold for all the 7 characters that we saw, that in case of hybrid, you have two different alleles, and only one is dominant, only one shows its effect. Okay, so this is Mendel's second postulate, that one allele tends to dominate over the other. So again, let's go back to yesterday's figure and see the F1 hybrids that we talked about yesterday. Was first F1 hybrid was coming from crossing purple flowered plant with white color. So you had capital P and small p, and since purple color allele is dominant over white color, you see the flower color as purple. Similarly, in case of cross between tall and short, you have in the hybrid you have one tall allele and one small allele. Only the tall allele shows its effect, thus making the plant tall. Is this clear, everyone? Okay, one clear. So now how does the recessive allele show its effect? The recessive allele, since it is weaker, it cannot show its effect when the dominant allele is present. The recessive trait you only see when the recessive allele is present as a pair. So when you have small p, that is both the alleles in a plant are white colored, then only you see the white colored flower. Similarly, in a plant, when both the alleles are for short height, then only you get the plants which are short in height. And this is again very easy to understand. The parents that we started with, they had the dominant alleles. The whole gene, both the genes or both the alleles are dominant. So you obviously see purple color, or you obviously the tall. So the point to be taken here is when you have different alleles in a single plant, only one is going to show its effect, and that one we are going to call it as a dominant gene. Okay, so to understand this process and really figure out why this 3:1 ratio, let's revise how actually fertilization happens, because then that will make it very crystal clear. So we know that when flowers are made, a plant makes gametes, okay, which are also called as sex cells. So in flower, you normally have both types of gametes. There is male gamete in the pollens and there is female gamete in the ovule. So this is how you see it. So normally in pollen, the male gamete is present, and in ovules, there is female gamete present. Now remember, there are thousands and thousands of pollen grains produced in a flower. So there are going to be thousands and thousands of male gametes. Similarly, in case of pea and many other plants, there are multiple ovules in a single flower. So each ovule will become a seed. So there are going to be multiple female gametes here in a single flower. Okay. So the significance of this will become clear to you shortly. So when you think of a plant or any other organism for that matter, all the normal cells in any given organism have a pair of genes for every character. Okay, so the plant could be having capital TT alleles, they could be having small TT alleles, or they could be having capital T and small t, that is tall and short allele. So all normal cells will have two alleles for every character. One is coming from one parent and the other is coming from the other parent. But when this plant forms gametes, something different happens. So when gametes are formed, actually what happens, each gamete gets only one allele out of the pair. So let's say the parent was capital T, that is tall allele. When the gametes are formed, the gametes will have only single allele for plant height. Okay, so this is what the male gamete will look like. This is what the female gamete will look like. So what really happens in fertilization? The pollen will bring in the male gamete. Now let's talk about male gamete will bring in, let's say, capital tall allele. Now let's talk about the F1 hybrid that we made yesterday. So the ovule, that is the second parent, is actually the short one. So the male gamete contains capital T tall allele and the female gamete contains the small t or the short allele. So when this male and female gamete will combine to form an embryo, now this embryo is going to be the hybrid embryo and it will have the tall allele and the short allele. So again, the gene pair is restored and this will go ahead, form the seed, and then the seed, when it germinates, will form a plant. So normal cells in a given parent have two copies of a gene. When gametes are formed, only single allele is passed to a gamete, and then when fertilization happens, the offspring gets back. Again, the whole pair is restored. Okay. Now, does anyone have this question that why is this necessary? Why do we need to do half? Why not simply have the gametes like the any other cell? Why can't gamete have both the copies of allele? Because it's actually very simple to understand that if the gametes had a pair, then one parent brings in two copies, another parent brings in another pair. So actually in this case, the offspring would have got two pairs, that is four alleles. And when this offspring becomes a plant, becomes a flower, and then forms gametes, the next generation would get four from one parent and four from other parent, making it eight. And this will just not work. A cell won't be able to function like that. So this is why in all organisms that reproduce sexually, gametes, when they are formed, they contain only half of the genes. So only one allele goes to one gamete.
We can also add that actually nature wants mixing. So, I mean, the parent gives only half. Okay, only one gene one parent gives, the other gene is brought by the other parent. So proper shuffling also takes place that way.
The shuffling is something that is caused by something else. So
I know that. I'm saying that it's like a parent gives half randomly, and the other parent brings other one gene randomly. Each parent brings one gene randomly. So what versions mix up is also decided randomly, isn't it?
yes, kind of, but it gets complicated actually when we talk about chromosomes, so we'll not go there. So basically the idea, primary idea is to keep the size of the genome constant. So shuffling can happen by multiple mechanisms. Okay, so actually when we talk about pure breed plants, we really get only one type of gametes. So since both the alleles are same, both the gametes, all the gametes in this case are going to be identical to each other as far as the height of the plant is concerned. Same is the case with the pure short plant, because it contains both the alleles are same, all the gametes will be the same. But when you come to the hybrid, it is containing two different alleles. So in this case, when gametes are formed, now although we are showing one only, that is we are showing only two gametes here, one of each type, there are actually thousands of male gametes in a particular flower. So please remember this fact. So in case of a hybrid flower, when the gametes are formed, 50% of the gametes will have the tall allele and 50% of the short plant will have 50% of the gametes will have the short allele. So if you look at number of male gametes present or number of female gametes present, in case of an hybrid, 50% will have tall allele, 50% will have short allele. So what happens in fertilization? Now we have talked number of times that fertilization is a random event. So what is happening actually, if you think about the pollen bringing the male gametes, so let's consider very, very simple. That is, the male gametes are from the tall plant, pure tall plant. So there is only one type of allele. Both the alleles are same, capital T, capital T. So both the gametes are identical, and we fertilize it to a female gametes which are coming from short plant. So here female alleles, female gametes are also having identical composition. So what happens is this male gamete can fuse with this short one, so tall short. Again in this case T and small t, tall short, tall short, tall short. So this is what is happening in your F1. That is, the hybrid that you create is containing one tall allele and one short allele. So you are getting all types of offsprings that are going to be formed. All of them will have one tall and one short allele. So that is why they all, since tall is dominant over short, you get all tall plants. Okay, so now let's do a matchstick activity, which will really make it very, very clear to every one of you, and then we'll come back and go further. Okay, so to Apurva, to you.
Am I audible?
Yeah.
Okay. Good morning, everyone. I hope people who were present yesterday, a lot of you are also here today. I can see a couple of similar names. And I'm also hoping that you have matchsticks with you right now. Matchsticks and also a couple of bindis in different colors. 2 different colors. So can you quickly respond on the chat if you have these things with you? I need to know from you before we start. Okay. All right, great. So yesterday we did a matchstick activity in which we were able to see what the plants look like, right? We used one matchstick to represent a tall plant and we used one matchstick to represent a short plant. Wait, I'm just going to put this in spotlight. Just give me a moment. Okay. All right. Okay, so let's begin and we'll start with our pure breeders. So you remember yesterday we knew that Mendel created, we talked about how Mendel went on creating a pure breed for different characteristics. So right now we are going to do this activity with just one characteristic, which is the plant height, just like we did yesterday. So we know that the tall matchstick, what we're able to see, the tall trait here, this actually, if you can tell me what is the hidden gene that will be present in all the cells of this matchstick? What is it inside the plant that is making it tall? And this is a pure breed. Can you reply on the chat? Yes. So we know that in all the cells of this plant, there's a gene, and I'm going to, again, just like in the presentation, I'm going to represent it by capital T. And you remember what capital T stands for? It stands for the dominant allele, right? That gives tallness as the trait. Now, at the same time, we have another pure breeding plant, but this time the trait that is visible to us is shortness. Now, this is a short plant. So what can we say about its gene pair? What kind of absolutely. Right? It has small t, that is the pair. So this causes this plant to be short, and this dominant gene, both genes being dominant, we can see that this turns out to be a tall plant, and here both are recessive. So that is the only, there is no case of one allele being dominant over the other. Both of them are recessive. So we see shortness as the trait. Now, what did Mendel do next? He crossed the two, right? And like Swapna ma'am told us, when crossing happens, So this is the representation we used yesterday. We have a box. We have the first half of the box represents the male part and the other represents the female part. And here, since we have two, I'm going to draw two. And again, remove the male part of one. Right? Now, we know that this is getting transferred from one plant to the other, but at the same time, when we are talking about cross-pollination here, we are talking about gametes, right? So these cells are not, okay, let me not use the word cell, but what Swapna ma'am used, gametes, which are essentially sex cells. So now I'm going to do something on these matchsticks to represent those gametes, right? So here, what about the tall plant? What can you say? You remember we spoke about that only one allele is passed to the gamete. So each gametes will not have a pair of allele. They will only have a single allele. So what is the possibility for the male gamete to have here? What allele can the male gamete have? T. Absolutely. There is only which is T, and I'm going to represent that using a black bindi. And I strongly urge that those of you who have matchsticks and bindi to do this yourself. So I'm going to place.
Apurva, I mean, like side by side, you can keep the full cell and show the gamete next to it, so that it's very clear that full cell had two.
You mean side by side as in?
I mean, just
okay.
Yeah, keep another matchstick here.
You mean using bindis?
Yeah. So a matchstick with two bindis to say that that was the cell from which the gamete was got one.
All right. Okay. So here, so again, this I'm going to represent using bindis again. So I'm going to put two black bindis. Somehow my spotlight went off. Okay. Yeah. Right. And the short one similarly has two red bindis. Right. So this is what the boxes, they represent what all cells of the plant have. And on the matchstick, I'm only going to talk about what the gametes actually have. Okay. So again, going back to my tall plant, here the male gamete has only one option. It will get only black. So what about the female gamete? Is there any other option? Okay, I'm talking about the tall plant here, Satish. Tall plant. And remember that this plant is receiving alleles. So the male and the females are receiving alleles from this box, right? Same T, exactly. We'll have a black only, right? So this is actually going to, oh, the bindi is stuck on my hand.
Apurva, not on the same match. If you are showing gamete, if your matchstick is gamete, then
I will remove it. I will remove it from here. I will remove that.
This becomes confusing. I mean, like you were saying, male has one, female has one, but if you put both on the same, then you have to keep two matchsticks.
No, so you can think of this as representing, you know, the bindis are also representing the same, the flower, right? Okay, let me just once move ahead, and then I'll just show what's happening next. And with the short one, right? Now here again, I'm going to put, yeah, red, and this one again will have another red, right? Now, what happens when crossing is happening? So when crossing happens, this tall plant, right? If you remember, let's say we are taking one black, the male comes from here, and since this is the gamete. I'm going to represent this within the circle, right? When they are combining. So this comes here, and now both of them, both these gametes will combine. Now these are from two different flowers. They will make an offspring, right? And each of that offspring, what will the combination be here for all the cells of that offspring? T and okay, one capital and one small. So again, I'm going to put, right? So all the cells, they get this, and I'm also going to write this representation. Here. Okay. So, but now if I talk about what this plant will look like, can you suggest which matchstick I should use to represent all the plants that are being produced by crossing these two? Tall. Absolutely. Does everyone get that? We are only going to get tall. And why is it that we are getting tall? If you could also answer that for me. Exactly. Somebody has answered. Prasida has answered that T. So the black is dominant over the red, which is why we see that, and black is the trait for tallness. So since the tallness trait is dominant over the shortness trait, the output, the plant grows up to be a tall plant, right? And all the cells of this plant will have this combination, one capital T and one small t, right? And we don't have just one plant, of course. I can use as many matchsticks to represent this tall character, right? Just as we did yesterday. All the plants that are coming by crossing the tall and the short plant have this character. But what is happening inside the plant when I talk about when the flowers come, right? So when the flowers come, we spoke about how they have a male part and a female part. And again, each of these, so the male gamete is going to get only one allele. So what could that one allele be? The one that the male gets. And I'll just write, this is our F1 hybrid generation. Okay. And this box here represents the F1, right? So F1 hybrid, all of them have a black and a red bindi, or a capital T and a small t, right? Okay. And again, I received the response on the chat that it can either get a black bindi or a red bindi. So let's go with the black bindi first.
Apurva, just show these tall plants with having both the genes here. So have two bindis on each of them and then do the crossing. Otherwise, once again, if you
I was going to actually show the selfing when it happens and then the combinations.
But right now, I mean, because it seems like the sticks are representing a tall plant. So it's not like, if you directly treat it as, say, what your sticks are representing is not very clear. That's why it's becoming confusing. So
the physical, what is visible is being represented by the sticks, which is
so these are tall plants, and they must have
all tall plants.
So they must have genes inside. So you should show the genes with two bindis.
Yes, so the gene, this box over here, this is representing the pair of genes that is present in all the cells.
I'm saying
the bindis that I'm putting on the ones. So I'm just showing that so that the what the gametes are getting becomes clear.
No, so gametes you can make in the next stage. No, I mean right now these are 4 tall plants or whatever number of tall plants, each having two genes. I'm saying that stage must be shown before gametes are formed. Otherwise, it's kind of once again confusing.
Okay, so I was using the bindis on the matchsticks for a different purpose, and all the cells I'm showing.
Yeah, that's why it's so confusing.
I would like to ask, okay, is it,
Wait. See, gametes are being shown by rounds, right? So you have showing them by round circles. And
yes.
if the C and gametes are also being shown by sticks, then it's like two symbols for
No,
the
the
same.
gametes are not being shown by sticks. Just the bindis on the sticks are showing what's happening in the flower, right?
So right now, all I'm saying is show two alleles on every plant.
I will do that, but it's going to change the process that's running in my head. You know, the representation I'm using it for. So I'll do what you're asking me to do, but
there's a confusion coming in. That's what I'm trying to point out. There's a confusion coming in because what you are representing is not unique. That's why there's a confusion. See, in when we use a concrete object, we have to be very clear what that concrete object is, right? So if this is a tall plant, if the stick is a tall plant, then this tall plant has
right now, okay, so I'm going to change the representation, and now I'm going to show what all cells of the plant contain on the matchstick itself. So all the F1 hybrids that we are getting, they'll have one black and one red, right? Okay, so whatever, all the tall ones, they're going to have similar pairs of alleles. Okay, so let's use just these many. All right. So what did Mendel do in the next stage? He, can someone reply on the chat? What was the next step? So far we have created an F1 hybrid that consists of all tall plants. Right. So Mendel self these plants. Okay, so I'm going to take one tall plant and another tall plant. Okay, so now he has self these tall plants, but right now, what are the possibilities for the male gamete? If I talk about just that, and I'm going to, okay, just give me a moment. Okay, yeah, so the male plants. Sorry, I meant the male gamete. It can either get the allele that's responsible for tallness, or it could get an allele that is responsible for shortness. And similarly, what happens with a female plant, it could also get an allele that's responsible for shortness, or it could get an allele that's responsible for tallness. Is this clearly visible? Yeah.
Yes.
Okay. So now when these gametes fertilize, and remember that these are just possibilities, right? What all is possible? So the male gamete can get the black bindi or the red bindi, and the female gamete can get the, again, similar, red or black. Now, when the fertilization happens, what are the combinations that are possible for fertilization to happen? So either this black and this red combine. Okay, and in that case, we get a pair of alleles that look like this. Wait, I need more black alleles.
You can use sticks for doing this representation.
Okay. I'll do that. So one black and one red. So what is the outcome going to look like? What's the visible trait going to be? Tall. Yeah, we know that there's one dominant present over one recessive. So the result will be, so a tallness trait will be dominant over the shortness one. And what happens when? Okay,
you can stick
let's
the two
do this
trees.
black.
You
so
can stick the two trees on the stick itself. I mean, then it's clear that it's what you're seeing is tallness, but inside there is these two alleles.
Okay. And now here, this black could have also combined with this black, right? And in this scenario, what would the outcome be? Again, tall, yes. So I'll need, so we'll put two black bindis. Okay. And the outcome is tallness again. Now, another possibility could have been that the male gets the recessive allele, which is the allele for shortness, and the female also gets that. Right? So what? Short, absolutely right. So both the new plant that comes, it has right? Okay. And Satish, let's take this question. Let's just finish with the last one and we'll come to your question. Okay. So now from all the tall F1 hybrids, suddenly it's very clear that, you know, we can actually see that one short has appeared, and what's happening inside that plant, that short plant has now appeared in the next generation. So all these new plants that we are making, they actually belong to the second generation, right? Another possibility was that the red from the male could have combined with the black from the female. Even that could have happened, right? And here again, many of you have already responded on the chat. Since we have one red and one black, we'll end up getting a tall plant again. And I need one red still. Right? So starting from my pure breeders, I end up to the second generation, and I'm able to actually visualize the entire process that's going on within these plants, you know, with the help of these really simple objects. So the request here for all of you is to actually make children do this, because this is the concept, you know, the theories that Mendel came up with, they are very mathematical in nature. And these objects, when you can visualize that math for them, it becomes really powerful, and maybe it'll be something that they can keep with them for life. You know, at least they can retain that concept, that why is this trait of shortness that disappeared in my F1 hybrids, it suddenly appears back in the second generation. All right, I hope most of you did this activity on your own, and now we can take any questions if there are. There was one question. Will it be more clear if we represent the alleles on two different chromosomes? Okay, Swapna ma'am, would you like to take this question?
Yes. So basically, Satish, we are just making a very simplified diagram. So if you add chromosome in it, then it just adds another layer to it without really clarifying anything. So because we are looking at only one pair of alleles, well, you don't need to have chromosome to get the understanding correctly. That is my opinion. So whether you have draw a chromosome there or not draw the chromosome. So generally in case of these genetic crosses, we don't add chromosome because that doesn't really solve anything. So what we are looking at is we are just focusing on alleles getting separated into gametes and coming back in the embryo.
And let me also add, I mean, when like what I was insisting with Apurva also, that if you are showing it a concrete object, it should become very clear to the viewer or doer. I mean, the child would be doing this himself or herself. It should become very clear what that thing is representing. If you have like too much complexity, this is a chromosome, or it is gene, or it is DNA, or it is allele, I mean, it's going to be messier and messier. See, then messiness already exists in theory. We are using concrete objects to actually clear some of that messiness. So there should be a very clear, precise definition of what the matchstick is, what a bindi is, and where it is located. And that's why if you notice, see, in Apurva's representation, she is using circles for gametes and squares for cells. That's also deliberate to be able to separate out which are gametes and which are cells. So I mean, even figuring this out requires, I mean, very not only clarity from the point of view of the thinker or the teacher, clarity for the viewer as well. Otherwise, viewer is likely to get even more confused. I mean, abstract ideas are anyway confusing. Mahesh has a question.
So Sapna ma'am, there's another question. If sex is determined by a dominant allele.
So Mahesh, actually, sex is not determined by a single allele. So there are multiple genes because there are basically a lot of characters associated with sex. So actually, there is a chromosome which is responsible for determining sex of an organism. So it's not a single gene. So basically, in case of males, normally in case of human beings, whether you have a Y chromosome or not decides whether you are going to be male or female. So it's not a simple case of it's just the presence of a chromosome or absence of a particular chromosome which decides the sex. Did I answer your question?
Just adding to what she said, I mean, even if you look here, right, you can say that why is it that we have only tall plants or only short plants, right? Or why did Mendel choose height as a character for his experiments? We've spoken about that he chose only 7. So I'm wondering if this part is also clear that from this, after we've spoken about genes and alleles, that why these 7? So, okay, I'm hoping I did not get any response. But if you see here, height as a character has the presence of only 2 alleles, right? So there is a capital T and there's a small t. There is nothing else that is present inside the plant, right? And yesterday we'd even spoken of blending a little, right, about this being a range. But when it comes to the plant height, it is not a range. There are only 2 possibilities, tall or short.
So Satish has a question. Will the student think that only 2 alleles for a character are present in the whole organism? As a matchstick, yes, matchstick represents a tall plant. tall, the whole matchstick represents a tall plant, Satish, but basically, if you're talking about a character, there are obviously only two alleles for a character in one cell. So if the plant contains 10,000 cells, each of these cells will be containing a pair of alleles for height. It's not that one pair for the whole organism, one pair in each cell of the organism. Did it make it clear for you? Okay.
I think this needs to be clarified to students many times. I mean, so that they don't have such misconceptions. That's why process is also important. How, I mean, where are genes present, for example? They are present in each cell. You don't say that the whole organism has one pair of genes, right? So it's not, see, what we are saying is that the activity does not replace all of the theory that needs to be clarified. Now, Mahesh is another question. Swapna ma'am, would you want to take?
Yes. So, yes, there are two types of alleles. No, in one pollen, there will be only one male gamete. Okay. But in a flower or on a particular stamen or in a particular anther, there will be thousands of pollens. Each pollen contains one male gamete. So one male gamete will have only one allele. But if you look at the flower as a whole or an anther as a whole, so 50% of the pollens containing male gametes will have one allele and 50% of the pollens will have another gamete. Okay, one pollen only has one male gamete. But if you look at all the male gametes in a flower, 50% of them will have one allele and 50% of them will have the second allele. Is it clear? So should we resume? So let's just briefly recapture what we did in the activity. So what we started was the parental generation. So what we took is tall plant and a short plant, right? Tall plant, which was a pure tall, so it would have both tall alleles, and short plant, which would have both short alleles. And then when the gametes are formed, since it has only same type of allele, it will form identical types of gametes. That is, all gametes will contain capital T in case of one parent and small t in case of another parent. So since we are starting with pure breeders, all the gametes for height with respect to height will be identical. So when these gametes are made and when you allow them to fertilize, you get F1 generation in which the tall allele and the small t allele will combine through cross pollination, giving you an embryo which is now a hybrid because it contains a different set of alleles, one for tall and one for short. So since tall allele is dominant over short, the plants, if you look at it, they will be all tall. So this is our F1 hybrid, which is has different alleles, but if you look at their height, they all look identical to the tall parent. So trying to trace where the short allele is gone, we basically do self-pollination of F1 plants. So if you look at the F1 plants, if you look at their allele composition, you have different alleles, one for tall and one for short. So when gametes are formed here, actually you will have two types of gametes being produced. So 50% will be having the tall allele and 50% will be having the short allele. And same is shown here. Basically, what we are so why we are showing you two here in case of, even if we are calling it self-pollination, is because you can consider this part as coming from the pollens, that is male gametes, and this portion. So there are going to be two types of female gametes in one flower, and there are going to be two types of gametes in the pollen grains. So when fertilization happens, either this could combine with this or this could fertilize this. So thus there are multiple possibilities. So when the F2 generation happens, so you are having two types of gametes in the pollen and two types of gametes in the ovule. So what happens is, a male gamete containing tall allele, if it combines with the female gamete in the ovule also, which also has the tall allele, you will get a plant which has capital TT, both are tall alleles. But if the male gamete with tall allele combines with the female gamete which has the short allele, you will get, okay. Now if this is the male gamete and this is the female gamete, can you tell me what will be the outcome of this fertilization? What will this give? Male gamete with small t fertilizing? Yes. So it will be a hybrid small t capital T. And the last one, where the male gamete is also bringing short allele and the female gamete is also bringing small allele, what will this give you? The last combination, this coming, yes, Rasida has answered. So what you will get is small t small t. So this is what we will get if we look at the alleles which are actually hiding from plain sight. But if you plant these seeds, what will you get? What kind of plant will you get? Can you tell in the first case, what will you get? This got revealed before you could get a chance to answer. Okay, second one, can you tell me what will the plant look like, tall or short?
Ma'am, this is a question by Lina S.
Yes, actually, whether you write small t capital T, why we have written it like this here, simply we are following the sequence. Because sometimes with kids, it is easier for them to understand because we are writing one from here and one from here, one from here and one from here. So maintaining the same sequence, if you look at the appearance or the effect, it doesn't matter which one you write first. So this and this is going to be same. Okay, Mahesh has written 3 capital T and small t. Maybe a spelling mistake.
No, but he's 3 capitals and 1 small.
Yeah. Mahesh,
So
would you like to elaborate as to what you're trying to say? So according to iPad and Preeti, both of these, this is going to be a tall plant. This and this are identical. Just we are maintaining the sequence. Okay. Yes. To be tall, and this last one is going to be. So this actually explains the 3:1 ratio, what we see, that is 3 tall plants and 1 short plant. But if you actually look at the genetic composition, or if you look at the alleles, you could write the ratio as 1 tall as to 2 hybrids is to 1 short. Okay. So if you look at the allele composition, it will be 1 capital T, 2 capital T small t, and 1 small t small t. So actually, the 3:1 ratio, allelic wise, is 1:2:1 ratio. So this basically what we have discussed in this activity covers 3 postulates of Mendel. The first one talks about each trait being controlled by a pair of genes. Second postulate talks about that whenever you have two different alleles for the same character, one allele dominates over the other. So you see only effect of one. And the third is called as a law of segregation. That is when gametes are formed, the pair of alleles, the alleles separate from each other, each gamete receiving only one allele. Okay. So of course, Mendel continued his work and then he work with two pairs, that is two characters, that is called as a dihybrid cross and further on. But in this initial first two sessions, we don't have time, so we are not going to discuss this his further work. But what we want to show you briefly is what has happened after Mendel. So once Mendel's work is done,
Ma'am, please take questions and then Mahesh, what he wanted to say, or if he has any confusion. So, I mean, I think it will be good to take it.
okay.
So, particularly, I mean, let's unmute Mahesh because he gave four versions, no? So, Mahesh,
I think what I understood it as the whole answer he gave in one. So,
or maybe.
Huh.
Mahesh, would you like to say? I mean, you just gave the whole answer in one go, or you
No, that
of?
was iPad. What I said.
Okay. No, iPad gave, but Mahesh had written, no?
Yeah, Mahesh. Yeah.
So, Mahesh, would you like to say what you meant?
He said, I have understood.
Yeah. Okay.
Okay.
Any other questions? If there's any other question or confusion, we have only a short while available, but anyway, if there's any confusion, then we should take. Yeah, so I think you can continue.
So what has happened after Mendel? So basically, we have come a very, very, very long way after Mendel. Yesterday, we briefly talked about that Mendel's work was not really understood when it was published because that time it really did not make sense to most people because cell was barely being seen at that time. But if you actually look at what has happened after Mendel, about 15 years later, nucleus was seen very clearly. Then inside the nucleus, people has started observing structures which they called as chromosomes. So if you this happened in 1880s, that different individuals actually saw structures inside which they called as chromosomes. And then slowly they figured out that it is actually chromosomes which are separated from each other during gamete formation. So they realized that genes, what Mendel was talking about, must be present on the chromosomes. So for next few decades, we understood what chromosomes are, and then in 1950s, we understood that chromosome is actually made up of a macromolecule called as DNA. So gene, as we speak of, is basically a part of the chromosome or a part of DNA which has a particular function is what is gene. So chemically, if you speak, gene is basically a piece of DNA, and this piece actually comes from a chromosome which is a big enough. So one chromosome will have many genes, and each organism has many chromosomes. We get one set of chromosomes from our mother, and we get one set of chromosomes from our father. So even in case of plants, you get one set of chromosomes which gives you one set of genes from one parent, and second set of genes which comes through second set of chromosomes comes through the other parent. So we have moved from an abstract concept of factor or a gene to now we are able to actually know, we get understanding of each gene per se. So when we say that there are two alleles for every character, so actually we get, let's say we get one chromosome from one parent and the second chromosome for second parent, and we can see that just like we have specific addresses for our home or office, just like that, genes also have addresses or specific positions on a chromosome. So if you look at this particular chromosome, if at this place there is a purple colored allele present, on the another chromosome which came from the other parent, you will have exactly at the same position, you will have the corresponding, that is the second allele for white flower. Okay, so genes have specific locations on chromosomes, and we have a set of chromosomes which have thousands of genes. Now we have reached the stage that we know sequence of each gene, so on, so forth. The next slide, this just because someone had asked me a question, I just wanted to say that after Mendel's work, actually these studies were repeated in these set of plants and these set of animals in next few decades. And this 3:1 ratio or his other ratio, which is dihybrid ratio, these were validated in beans, sunflower, cotton, wheat, barley, tomatoes, maize, so on and so forth, as well as in cattle, sheep, cats, mice, rabbits, guinea pigs, chickens, pigeons, canaries, and moths. So Mendel's work really has been valid in almost all systems tested, so it is considered a universal. So very briefly, I just want to talk about that. What about human beings? We can't do similar experiments like we did with peas or mice or maybe honeybees, so on and so forth. So how do we study genetics of inheritance in case of humans? So basically what is done is you tend to record features that are seen or traits that are seen in families and then do the analysis. Okay, so I just want you to be aware of certain, just like in case of pea, Mendel used either flower color or height of the plant or color of the seed. Like that, there are quite a few easily seen features in case of humans which also show this kind of behavior, that is they are they are caused by single gene. So one of the simplest characters that everybody can study, all of you can check yourself now, is can you roll your tongue like this? So this is also a dominant gene, dominant allele, people who can roll their tongue or people who cannot. Okay, so please try it out and put it in chat box. And the similar case is if you have gone and looked at your family members or friends. Yesterday also we talked about this, that how many family members you could find with either of this phenotype. Okay, so if you've been able to find that all are same or all are different, that will be very interesting. So these are many features, and there are many features like that which we can see and analyze. So you can use many of these features in a classroom when you talk to students. So this is called as a widow's peak. You have a triangle, the hairline is different, so this is dominant over this. We already talked about earlobe. uh, okay, I will just take the question of Lena in a second after this slide. So just because we talked about genes, we talked about a pair of genes. So I just wanted to put these things in perspective, that when we talked about genes, actually, if you see that different organisms have different number of genes, and this just slide shows you that a typical bacteria has about 4,000 genes. If you look at a fruit fly, it's about 13,000 genes, and humans have around 31,000 genes. But if you look at P is actually quite complicated, has more number of genes than even humans. So this is just to put in perspective that there are so many genes in every cell, and then in pairs, and that is how they are controlling all the characters. Now let's start with Lena's question. So she has asked why we don't consider nucleotide sequence in parental dispute. See, the thing is, Lena, that actually, if you want to sequence the whole genome, that would be sequencing 31,000 genes. And that is just too much, too expensive, and too time consuming to do this in case of paternity testing. You don't really need to do that. But there are certain markers that which are much easier to figure out. Okay, so that is why in DNA fingerprinting, you basically check for certain markers. And if you see the same pattern as whether you see the same pattern as their mother and father or not, you can do the paternity testing. Now, Mahesh has asked, how do new qualities arise then from parents? New qualities arise because, as Anshumala ma'am also just mentioned earlier, there is lot of shuffling and lot of changes that happen. So I just wanted to show you this image. Just don't go by absolute numbers. But when we talked about random shuffling of genes, so actually when we have 23 pairs of chromosomes, when they are being shuffled, one gamete gets half and the other gamete gets the other half. Every time a gamete is formed, random separation of chromosomes happen. So if you look at actually one individual, there are totally so many different types of gametes forming in a single parent. So there are that many combinations of genes that can happen. That is how all the siblings are not identical of the same parents. And what is hand clasping? So hand clasping is basically if you are not thinking and you clasp your hand, and some people tend to put their left toe, left thumb over the right thumb, or some tend to do the reverse. So these are simple kind of characters people have noticed that you see as differences in different individuals. So there are many characters like that. There are, but this has to be done without thinking. If you think and do it, then everybody will try to do it in a certain way. So when you're not thinking, when you clasp your hand like that, you have to see which thumb is above, whether the left thumb is on top or right thumb is on top. So this just gives you interesting points to notice about different individuals, different variations that you can
Ma'am,
see.
Mahesh had asked that other than parents, how can new qualities arise? So I don't think you right now taken that question. And even Lena was asking in paternity disputes, why is mother's genes not or mother's nucleotides sequence not considered? So the question was
Because
different.
you are trying to decide who the father is. Why will you consider mother's DNA? 1 second, let me just show you the screen. Are you? Huh. Okay, let's see. Forget the details, but let's just think this is father's gamete and mother's gamete, and a right. So let's say this is what is the offspring whose paternity is being tested. So what you want to see is you take the father's DNA and match it and see whether half of the offspring's DNA is coming from that particular father or not, because the only paternity is being tested. Okay, because mother delivers a child, so one can be almost 100% sure that this child belongs to this mother. But in case of paternity, only 50% of the genes you have to ascribe whether it is coming from the father in question or not. That is why you will only test father's DNA. But if there is a doubt that both the parentage is being in dispute, then you will have to check mother's DNA. But normally, if there is complete documentation available that a particular mother gave birth to this child, you are not doubting mother's DNA. You are only considering father's DNA. So it is only sufficient to do father's DNA analysis. And in that also, you will do this done by DNA fingerprinting, simply because so DNA fingerprinting has nothing to do with actual fingerprint of on the finger. And Mahesh's
Mahesh,
question, how do new qualities arise other than from parents? Is they are not arising other than parents. Okay, but 1 second. So actually, let's see, if you're talking about humans, we have 23 chromosomes coming from mother, 23 chromosomes coming from the father. So let's say these are the two homologous chromosomes. So what is happening is, first of all, there is random segregation of genes. Second is there is lot of exchanging. So there is portions of genes or portions of DNA are exchanged between the chromosomes. Then there are other mechanisms, which I don't know whether you are aware of, but there are these are called as jumping genes. So there are DNA sequences which basically move from one place to the other, and they create different sequences, or they basically alter a gene. So if a gene is altered, a particular quality will be altered. So there are a lot of different things that happen. It's not just simple dividing, just like we are dividing a pair, maybe a pair of a group of marbles between two children. It's not as simple as that. There are a lot of mechanisms which shuffle genes between a given cell before gametes are formed. This is what creates new characters. But I think that discussing molecularly what is causing all that might be too complicated for this audience. But you can read over crossing over and different mutagenesis to understand that there are hundreds of different things that are happening to create newer information. That is what is responsible for evolution. So a lot of variation is being caused, and multiple mechanisms which are actually important for evolution. So I think we can end here from my side.
Prasida has said that if we are doing online classes, Prasida, we do keep doing online classes all the time. So you can stop sharing.
Okay.
Prasida, we do keep keep doing online classes on a variety of topics: physics, chemistry, biology, and environmental science. Lately, we have switched more to specific groups, like your group today, where the audience is pre-decided. Because if we do random open classes, we are unsure about people's attendance, and often, I mean, a whole lot of people will register but not necessary. And since there's no charges, at least as of now, there are no charges, so people register non-seriously but not necessarily attend. So of course, I have talked to KSSP that we would like to do online classes for Kerala teachers if KSSP can be the coordinator. I mean, if KSSP can coordinate, somebody has to take responsibility of taking the information to people and ensuring that enough people come as audience. I mean, we have tried doing it through WhatsApp groups, but it doesn't necessarily happen. I mean, it doesn't necessarily ensure that we have a sizable audience for us to put in that much effort. Even today, if you see yesterday and today the number of people has dropped. We don't know the reason why. I mean, it could be a Sunday and people may have their other considerations, but we want to ensure that if we are not charging, this is sponsorship. Somebody is sponsored, but the sponsors finances and our efforts should be fully, at least judiciously utilized well. We wouldn't like to waste money and time. So that's why if KSSP is ready, we will definitely be happy to do classes for Kerala teachers. We are doing for some other states as well. And we do want to keep working continuously with some groups so that we can see more effect of these inputs. I mean, like there's sustained input, which leads to sustained benefit. And of course, teachers can get back to us saying what kind of problems they face in classrooms, what are the concepts very difficult to understand, and what are the concepts or topics where they want help. And with us, like constantly new design work goes on. So for heredity for us is a very new topic. We've recently, over last 1 month, we have worked. We will develop it even further beyond Mendel's contribution. And typically, one topic may take months for months of development, then trial with children, and then only it is actually offered to teachers. So that's our usual style of working, and like topics can get refined over years also. They keep getting refined, and we do support our topics with resource material which can be passed on to teachers, so that full help to teachers can be given for enriching their classrooms, their personal knowledge, their personal teaching techniques or pedagogy, and also reference material. So Lina is saying, please continue this type of classes. Thank you, Lina. If you say that this is helpful, yeah, we want it to be that way. We can post the updates in the same group. Instead, we would request you to join our general broadcast groups. Manisha, can you paste the link to the general WhatsApp group? We have created some broadcast groups where we broadcast our activities, where anybody can join. And of course, if we have specific webinars for Kerala teachers, I mean, with KSSP or any other organization, we will be very happy to offer you people. But typically, we wouldn't like to see, I mean, right now, amongst the 26 people, nearly, we are more than 6. Our own group is more than 6. Only 20 of you are present. We will not do workshops only for 20 teachers. Enormous effort goes on behind designing, and then of course offering. You can see there are always more than one resource person. So much effort and only 20 teachers respond is not our idea of good communication. I mean, you know, if this was a charged workshop, a typical company, or for that matter, and even an NGO, would charge heavily. We don't charge, but we want seriousness from the other side. And Manisha has posted, okay, Apurva has posted one link for a broadcast group, which you can join for updates, for information on any open classes that we offer. We do offer open classes. We did a lot during COVID. We offered a very large number of webinars, hundreds of them, but now we have made it less frequent. But we certainly want to work with organized groups of teachers, sometimes with children. Children we treat as our labs, where we ourselves learn pedagogy. So we offer sometimes with children, but our primary target group is teachers. So anything else you want to say before we end this session? Any suggestions? And of course, suggestions for future, or anything related to heredity that you would want in future sessions. Manisha has put in the link to our Facebook page, and Manisha, can you post the YouTube link also? So there's a YouTube channel as well. So we are trying that all our sessions are freely available on social media for people to utilize whichever way they want. Nashida couldn't attend yesterday, so she watched on a YouTube channel. Great, Nashida, that is the idea. People miss topics because of their personal engagements often, or at times because of internet or connectivity issues. So that's why it's told. Yeah. Yeah, please. Anybody wants to say something? You can unmute yourself if you want. So Manisha has also posted the YouTube channel link. So anything else anybody wants to say? We, because of you people, because of this Mendel celebration, we hurried up with our this topic, Mendel and Heredity. We put in special effort to do it fast, so which is actually good for us. So it did. So I mean, like I mentioned yesterday, Swapna ma'am, Apurva, and Assis Mansingh, 3 of them were working hard over this to finish it by yesterday and today. In fact, Apurva worked overnights, probably, almost. Will the class continue? Not this one. This was only for this Mendel's centenary celebrations. So Yeah, welcome everyone. So in case there is no other comment, so thanks for attending, and we were happy to connect to you, and I personally enjoyed these sessions a lot, these two. Yeah, welcome friends, and hope to see you once again in other workshops, other webinars.
Thank you all.
So Swapna ma'am, should I end then?
Yeah.
Okay.